Good afternoon, everyone. I hope everybody can hear me. Thank you for joining us in this first lecture series of Philippine Planktology. Before we begin, I would like to remind everyone of the common house rules in webinars. We are requesting all participants to turn off their cameras and their microphones on mute. And then please hold your questions until the end of the presentation. We will entertain those questions at the end of the forum. And then if you want to ask your questions live, please raise your hand and wait for the moderator to call you in. You may also type in your questions in the Q&A box, the chat box below. And also, we have FB Live right now on the Philippine Planktology Facebook page. You can also comment your questions there, and then we will have people working on collating all your questions. And then also at the end of the forum, kindly accomplish the evaluation form on the link provided in the chat box to claim your e-certificates. Thank you so much. By the way, I am Christy. I am a faculty member of Info CFOS UPV. I am also the Vice President of the Plankton Society of the Philippines and one of the admins of Philippine Planktology page. And also I'm your host today. We are bringing this webinar to you from different locations, including UPV in Miagao, Iloilo, from MSU IIT in Iligan, and Taipei, Taiwan. So good afternoon, everyone. Maayong hapon and tajahao. Okay, so to formally open the program, please welcome my colleague, Mark Louis Lopez, the most admin of the Philippine Planktology page. Thank you.
Good afternoon, everyone. So it's my pleasure to welcome all of you and thank you for joining us for the first talk for the first ever lecture series on Philippine Planktology. So the idea of having a lecture series for today, it all started with a simple conversation with a co-student. So Christy and I are both PhD students now here in Taiwan. And our primary objective is to invest as early as now to convince younger generation of people to join us in studying planktonic organisms, may it be zooplankton, phytoplankton, or the microbial planktonic organisms in any aquatic ecosystem. So, and slowly we're seeing it becoming a reality. And we're having different participants from different institutes all over the Philippines and in other countries. So maybe I'll have a short roll call of the participants and their institutes. So we have participants coming from the University of the Philippines, Visayas. Also, we have participants from Mindanao State University, IIT, Caraga State University, Bohol Island State University, Xavier University, the DOST people. And we also have participants from the University of the Philippines, Diliman. We also have participants from Cebu Technological University, Cebu Normal University, the Central Mindanao University, Mariano Marcos State University. Surprisingly, we have participants from Philippine Coast Guard Auxiliary and other participants coming from different institutions all over the Philippines. We also have participants from Japan and from Taiwan. So I'd like to welcome all of you. And at the same time, aside from welcoming the participants, we're very thankful for the support of our other organizers. Specifically, we have the Institute of Marine Fisheries and Oceanology of the University of the Philippines, Visayas, especially with the support of the Director Sheila Santander de Leon and Professor Jonaline Metillo. We're also collaborating with the Plankton Society of the Philippines and the Philippine Planktology page, as you all know. So without the help and all the effort of these people, this event will not be possible. At the same time, I'll also want to grab this opportunity to thank our speakers. So they are our mentors in the field. And when we try to approach them and ask for their help, they are very much willing to extend their expertise and share their experience to younger generation who hopefully invest time in studying plankton, and hopefully Philippine planktology as a field in the Philippines will be recognized. And I would like to keep it short. I hope everyone will enjoy, and thank you very much. Hope we'll have a fruitful discussion after. Thank you.
Thank you so much, Louis. Our speaker for today is Dr. Ephrime Metillo. He received his PhD from the University of Tasmania in Australia. He is currently a professor of Mindanao State University Iligan Institute of Technology, also the Dean of the College of Science and Mathematics at the same university. Dr. Metillo's scientific works focus on population biology and community ecology of marine plankton species that is relevant to ecosystem management. He has published numerous papers on taxonomy and ecology of a wide variety of planktonic taxa like marine phytoplankton, copepods, isopods, jellyfish, and ichthyplankton in central and southern Philippines. As a seasoned planktologist, he has mentored a lot of young researchers in the field of planktology in the Philippines. He is the current president of the Plankton Society of the Philippines, which he also established in 2013. Now, let us sit back and relax as he tells us about the Philippine zooplankton as wanderers and ecological drivers.
Oh, hello, good afternoon. So, everybody can hear me? Okay, then.
Yes, sir, we hear you.
Okay. So, thank you very much, Christy, and also thank you, Louis, for mentioning that the importance of zooplankton or the importance of plankton in our lives. Very important. I hope hopefully you will realize later after my talk. And okay, I'll go straight to the presentation. All right. So is it showing now? Christy?
Yes, sir, I can see your presentation.
Okay, so yeah, I also would like to acknowledge our friends from the Bureau of Fisheries and Aquatic Resources. No, I think I saw the name of Anna. She's also a member of the Philippine the Plankton Society of the Philippines. It's always Plankton Society of the Philippines for its name, for its acronym, it's PSP. And you know what PSP is. Some of us might know what PSP is. And also, I'm quite honored and privileged to be invited to present this talk. And I'm also happy that our friends from the Philippine Coast Guard is actually listening. It's good that we have several representatives from different sectors, no, working in our ocean and seas. Okay, so my talk is focused on zooplankton this afternoon. The title, From One Wanderer to Ecosystem Drivers. I hope you will develop some insight or perhaps explain to yourself why I developed this title. No? Anyway, we can discuss this later on. First, I'd like to give a little bit of an outline as to for the talk. Okay, so I'm gonna give a very brief backgrounder on the topic, and also I'll present some definitions and a little bit of classroom lecture on a framework that would actually focus or highlight the importance of studying more on zooplankton, specifically Philippine zooplankton. And I will also define what the meaning of ecosystem driver, and again, focusing on zooplankton. And after that, I will mention three main researches I have done in relation to the topic. So specifically, the discovery of a certain species of euphoseed or krill, true krill, their role in the pelagic ecosystem, specifically on specific large marine organism. And then a study on seagrass demersal zooplankton, and thirdly on the importance of copepods, or yeah, copepods in our sardine fishery. And then towards the end, I will pose a challenge. No, I hope our young students interested in studying zooplankton will take this up for their research interests, and that is to link zooplankton biomass or zooplankton biology and ecology to our fisheries. No? Our fisheries is very important in this country, yeah, specifically on how we can actually predict state or status of fisheries, making sure that our fisheries will be sustainable for a long time. So how can we devote our energy to study the link between zooplankton and fisheries so that our children, I think Eric has children already, so he should think about if our children will still be able to catch fish, or our children's children. So we are doing things today not for ourselves, but actually for our future, for the future of the country, for the future of next generations. And then, as a summary, I will highlight, uh, yeah, again the importance of zooplankton, Philippine zooplankton specifically. Okay, so well, I'm very thankful to Christy Acabado. She's actually the present vice president of the Plankton Society of the Philippines. This was established in 2014, and we now have about 166 in the audience, and perhaps more watching at Facebook. We encourage you to actually join the society. We aim to have a new election this coming conference for the Philippine Association of Marine Science in PAMS. And we hope to come up with a new set of officers that could actually continue on with the purpose of the society. You know, we have to keep the Plankton Society active always. And I'm actually very happy that Christy and another partner, I forgot his name, they established a Facebook website called Philippine Planktology, where everybody can just pitch in information and probably learn something from the postings in this website. So I'm very happy that this lecture is also broadcast through this website. And that was the main reason. The invitation was given to me, and of course, without reluctance, I accepted the invitation because it's a chance for me to share my very few limited knowledge on Philippine zooplankton. So we go now to the definition. We know the plankton. These are organisms found in water, no, or even air. So probably this is your first time to hear that plankton can also be found in air. No, so very specific example would be the eggs of copepods. The eggs of copepods can actually be carried in the air through marine droplets or aerosols, and they travel through. They can be carried by the wind and collect in bromeliads. No? So if you know what bromeliads are, if you're a plantita, you notice that in the middle of the plant, there's water collected in it. And if your bromeliad garden is actually very close to the sea, or if a forest with attached bromeliads on the trunk uh, will have water in it, and so those coffee pod eggs will actually be collected in these plants and hatch and become inhabitants of the water in bromeliads. So that's what plankton are. It says here that they are propelled by the wind and currents of the water. So in lakes also we expect to have plankton. And well, as most of us know, the term plankton is Greek. I wish it was a Filipino term, but someday some of you might actually develop some terms related to plankton. So it's a Greek word meaning planktos, meaning wanderer or drifter. So, and this was also emphasized by a European guy, Victor Hansen, in 1887. But later on, we now realize that the plankton, specifically zooplankton, are not just simple wanderers. They're very important. They're very complicated. They're very exciting sort of organisms. And these are all information from Wikipedia, so you can just click on this and have a read. And I also would like to remind our planktologists that there are several groups. We define them according to feeding or trophic behavior. So we know phytoplankton. These are the plant plankton. No? So composed of diatoms, cyanobacteria, dinoflagellate, coccolithophorids, and others with chloroplast in it. No? And then the zooplankton. These are the uh, metazoans. Phytoplankton are single-celled organisms generally, but they exist also as colonies. Zooplankton are the animal component metazoans, including single-celled animals or protists. So single-celled organisms without chloroplast. Then we also have, of course, the mycoplankton, the fungi, fungus-like organisms, bacterioplankton, our bacteria in the plankton, and virioplankton or viruses that are floating in aquatic environments or even in air. There are planktonic organisms with double life, so they're like AC/DC, pwede kuryente, pwede battery, no? So we call them mixotrophs. All right. So in the presence of sunlight, they can activate their chloroplasts, but without sunlight, they can turn off their plastids that trap light. So we call them mixotrophs. Okay, so what is an ecosystem driver? So if you have a car, you cannot run a car without a driver, of course. So very important of a driver uh, to move your car from point A to B. So, but ecosystem drivers are natural or human-induced factors that directly or indirectly cause a change in an ecosystem. So essentially, we also could term them as pivots. So basically, if you change this particular factor, then the other components of the ecosystem will also be changed. So specifically, processes in the ecosystem. And they can be direct drivers or indirect drivers. And I recognize, or I believe that zooplankton or the plankton are ecosystem drivers. In fact, phytoplankton, without phytoplankton in the ocean, I don't think the carbon dioxide in our atmosphere is being alleviated, or the excess CO2 is being alleviated at the moment. You know? So zooplankton as ecosystem drivers just mean that they are not simple animals, or not. They're very complicated organisms, multicellular organisms in our seas and oceans and in lakes. And proof of that, you watch, you click on this website and be familiar with Sheldon Plankton. And this is kidding aside, no? I realized when preparing this presentation that we have actually a friend called Sheldon Plankton. And the image is actually used by our Philippine Planktology website in Facebook. So try to meet Sheldon Plankton, and you will realize that he or she has a very exciting and complicated life. Indeed. Okay. So complicated. So a little bit of complex topic is the idea or the challenge to all of us, you know, to all planktologists. How do we link zooplankton to ecosystem functioning? No? And one of the function of ecosystems, we know that ecosystems give us services. And it's funny, we always make use of this service without even paying back. You know? And that's a very common situation. We are so dependent on our ecosystems in this planet, but are we paying back? Probably we spend our money uh, without realizing that we're actually destroying or negatively affecting our ecosystems. And one of these ecosystem services, of course, is the food that we get, the fish that we catch from our marine ecosystem. So there is a challenge on how there's still a big challenge on how we can link zooplankton and the ecosystem service, and one of that would be fisheries service. Uh, and in 2016, Hebert and his colleagues and her colleagues actually, or maybe he is a he, Marie Pierre, maybe he's a guy, but he might be French, and they proposed that for us studying on zooplankton, we shouldn't be limited our thoughts on just looking at the organism as it is, no, but know the species and knowing the biology of the species does not only focus on the individual members of that species, and not even just on the population, but on as a community, or at the level of ecosystems. Now, we know that in ecology, there are several levels, no? From the organism to the individuals to the ecosystem level. And the authors are guiding our analytical systems on zooplankton, excuse me, to address studying them at the individual or population level and community levels, uh, and then without forgetting that they also have a big role in at the ecosystem level. No? So, for example, if you look at the drawing in the middle, this is a typical herbivorous copepod. Probably this looks like a cartia, maybe. So a herbivorous copepod mainly feeds on phytoplankton or planktonic plants. And we don't look at this particular species as individual species, but actually we always have to consider their impact to the ecosystem. No? So we study copepods as a group of species, as a group of zooplankton, but also having in mind their impact to the ecosystem. So this illustration shows us that these particular herbivorous copepod depend on oxygen and phytoplankton as prey, And of course, they give back CO2 as respired gas. And then, of course, this population have outflows. If you look at these arrows, other outflows aside from releasing CO2, but they are food to predators. And being food of predators, they are very important to higher trophic levels, to other higher trophic levels in the ecosystem. So I'd like to emphasize that boxes here actually represent ecosystem features or ecosystem processes. We study the Acartia as a species, but we have to keep in mind that they have influence at ecosystem levels. So they can be prey to predators, and their excretions or waste materials actually become part of the chemical cycling in the marine ecosystem, which is participated by back to the phytoplankton because phytoplankton are dependent on nutrients and deposition of carbon to the bottom layers of the ocean. So in fact, we call them as a biological pump. So with the help of zooplankton, carbon that's absorbed by phytoplankton from the atmosphere actually shunted or delivered down to the bottom layers as carbon pump through sedimentation. And then, then you have also the zooplankton biomass. All species of zooplankton in the ocean aim for producing a zooplankton biomass, and that's driven by natural selection, of course. All species in this planet would like to dominate if possible. That's the dictate of natural selection. And this zooplankton biomass will be linked again to biogeochemical cycles. And one of the cycles of, but one of the biogeochemical cycles would be uh, the use of carbon in terms of fish carbon or in fisheries. So this is just looking at herbivorous copepod. You can replace this herbivorous copepod with other zooplankton species with different feeding habits. So you can have omnivorous or carnivorous copepod. But for future students in zooplanktology or in planktology, please be guided that we can actually have a new way of thinking or perspective that for us studying zooplankton, we might be focusing on functional traits, but we don't forget that the ecosystem functions of our zooplankton species. So this diagram here actually, uh, the main topics or the main items that you need to study basically based on energetics. Now, it is best to analyze the role of zooplankton with considering also the energetics aspect. No? So in terms of, I mean, you would have a more productive approach of studying zooplankton if somehow you will take this kind of perspective. So for growth and reproduction, you focus on growth rates, reproduction, the number of offsprings at the level of functional traits, uh, and then consumption of your species, predator avoidance and excretion and ingestion rates and metabolism. And then if you focus on these studies, you're actually also contributing to ecosystem functions of our zooplankton. Yeah. Okay. So basically, this is actually a horizontal illustration of the circular cyclical image that I have shown earlier, but this one is just brought horizontally. I hope you have appreciated this. Okay, now just going now to the specific examples based on the things that I have done in collaboration with some colleagues. No? So I just want to show you that zooplankton first would be the importance of zooplankton in our coral reef ecosystems. So very important. The reason why I place coral reef here as the first example because coral reefs are really are vital, no? Are vital to our country, to the life of our oceans and for sustainability and for the life of future Filipinos. So we know that our coral reefs are continuously being degraded. No, in fact, massive areas are degraded at the West Philippine Sea, and you know who is the culprit? No, but somehow they are not paying where they're supposed to pay uh, because of the damage. So this is a food web, no, an ecosystem representation of a coral reef. Very simple Bolinao reef flat representation. This was published in 1990 by Dr. Perry Alinio and colleagues of UPMSI. So one of the first food webs being published in the country. And if you look at the red circle here, it encloses the zooplankton component of the ecosystem. Now, these are mainly early stages of mollusks, no? The trochophores and the veligers. So basically, the Bolinao reef flat has well, used to have plenty of molluscan bivalves and gastropods in the reef flats. And so the zooplankton component there would be mainly contributed by their planktonic stages, you know, the veligers, and also crustaceans, of course. This is a zoea. And you know, just look at the different arrows and linkages of the zooplankton. And the message is if you remove this component, then I would expect that the rest of the components will also disappear, and specifically those dependent on zooplankton. So this one is again a trophic features of the Great Barrier Reef coral reef ecosystem. You know? And I've encircled here zooplankton component of the reef. So all these complexities of coral reefs actually go down to the zooplankton, go down to the basic components, the basic component of the ecosystem. And I think zooplankton would be very important, apart from these primary producers at the bottom. And I think, yeah, there's a picture here of a manta ray and even a tiger shark, no? Tiger sharks of Tubataha Reef, uh, I mean, Central Sulusi are actually dependent on Tubataha Reef also. No? I have some information from our NGOs working on tiger sharks. No? And having mentioned Manta Reef, we tried to analyze the diet of our Philippine manta. Very common is Mobula japonica. And we used stable isotope analysis, carbon and nitrogen stable isotope analysis, aside from looking at the stomach content. And we found out, so this is a two-dimensional picture of the two isotopes we collected from the Mobula japonica, from the manta ray and potential prey, based on their stomach contents. And we found out that uh, the manta rays are actually dependent on krill, no? On 80% or 90% of their stomach contains this particular species of krill. No, it's not alamang, but it's true krill, family. I mean, Euphausides, no, the other common name of true krill. And the krill actually are also dependent on the herbivorous or the smaller size copepods. You know? So you see here we have two zooplankton types. You have a bigger zooplankton krill and the copepods contributing to the diet of our mobulids. And then we also did a study on demersal zooplankton. So demersal zooplankton, these are zooplankton that somehow live very close to the bottom during daytime and emerge at sunset to shallower layers. And again, I'm going to show you a classic ecosystem illustration. This was done by Dr. Miguel Fortes, Mike Fortes, one of our pillars on seagrass ecology and biology. I think he's already retired. Also with UPMSI. And it's good that Dr. Fortes has already recognized the importance of phytoplankton and zooplankton in our seagrass beds. You know? And then from our study, we also realized that this simple illustration on the role of zooplankton is quite simple, and actually it's more exciting. So we looked at, again, using carbon nitrogen stable isotopes. We looked at the food webs of seagrass beds of Santiago Island, again in the reflux of Bolinao. And I'd like to give honor to Dr. Ronald Villanueva, a very prominent scientist. He already passed. Right after finishing this study, somehow Dr. Villanueva uh, is in heaven now, watching over us. Okay, so we looked at impacted sites. We know that in Pangasinan, in Bolinao, there's so many fish cages, culture of bangus, no? As fish cages, and we sampled the seagrass beds in that area. In this particular area here, because this is the outflow component, so most of the waste materials from the fish cages flow in here. I means impacted and U is unimpacted. We also collected demersal zooplankton and fish in the seagrass beds in this area, and we found out. So this is the carbon and nitrogen stable isotope projections. A here is the impacted area, and we were able to determine the isotope values, carbon nitrogen isotope for the feeds given to the bangus. And also this is the unimpacted side. But in illustration, we see here that in impacted areas where they put a lot of feeds on their very extensive bangus culture in that area, somehow the ecosystem there seems to be dependent on the feeds already, you know. So the feed seems like a primary source of primary production. So aside from the typical primary producers, algae, seagrass. Uh, so even our demersal zooplankton became dependent on the feeds, no? And very interesting, the diversity of demersal zooplankton was reduced. And we noticed also that the fry of the barangen or Siganus possesses danggit were very, very thin, and also, of course, very few in numbers, because of the polluted, degraded area. Compared to the unpolluted, unimpacted site, we still have quite a diverse collection of demersal zooplankton. Our danggit and other species of fish are quite, the juveniles specifically are quite very happy, very normal sizes. But in the impacted site, our fish there were actually malnourished. So it seems we theorize that because the fish in the impacted site are not getting the diverse collection of demersal zooplankton prey, they became malnourished. So they're quite thin and probably affecting their reproductive capacity, but we did not examine this compared to the unimpacted site. Yeah. Okay, then the third example that I'm going to show to you is our study in Zamboanga Peninsula, where we have our Sardinella lemuro, no, our most common sardines at the moment in the country. Yeah, these are our sites. This is in the northern Zamboanga Peninsula. We collected sardines and phytoplankton and zooplankton inshore and also offshore. and looked at and examined their stomach content and carbon and nitrogen stable isotopes. And this is a picture of our Sardinella limura, and this is the two-dimensional picture of our data, nitrogen and carbon stable isotopes. This cluster here are the values for sardines, and these are for zooplankton, and these are values for inshore phytoplankton and offshore phytoplankton. So we were able to see the classic dependence of our sardines on zooplankton. No? And then after looking at the stomach contents of our zooplankton, of course, we found almost all copepods. No? These are Temora species. I think Temora turbinata, or I don't know, but I forgot the species, but these are temorids. So these are calanoid copepods. So essentially the stomach content of our Sardinella limura that during our study is actually 80 to 90% copepods. And then from our zooplankton sampling also, we found that copepods dominate the samples, about 80% or 90%. And we were able to find a large species of copepods. We also we call it Rhincalanus nasciutus. The size could be really very big, about 2 to 3 millimeters. And we also studied this, and we realized I think this output, this publication is also co-authored by Christy and Willie Campos, and we noticed that these large copepods are quite abundant in northern Zamboanga Peninsula, keeping our sardines very happy. And our main conclusion was that there seems to be a vertical separation in terms of locations of different sizes of sardines. The younger ones are shallower, the adult are somewhat closer to the juveniles, whereas the sub-adults or the bigger juveniles are seem to be found to be at the deeper portion, but they're all found at the inshore portions. This kind of arrangements. That is because the inshore portions of this area would also have high concentrations of copepods. So, but I would like to tell everyone that if you really want to take care of our sardines, right now their stocks are still quite low. We're hoping that with interventions from the national government, our sardine stocks will improve. Dr. Campos, Willie Campos, if you want to know more about the status of the sardine fisheries, well, we have to talk to Dr. Campos. But what I want to highlight is that we have to take care of our near shore, our coastal environments, our bays. We have to love our bays. We have to keep them clean because that's where the food of our sardines are, perhaps other species of pelagic fish in our fisheries. These are the feeding grounds of our pelagic fish. And because most of our zooplankton actually bloom here. And if you have come across with the publication of Dr. Cesar Villanoy and his team at UPMSI, they were first to identify that this area here is actually a coastal upwelling phenomenon. Sir Gao, naapa ko yung webinar, kadyot lang. Bye-bye. So they were the first to actually discover that there's an upwelling event, a coastal upwelling. It's not very large like the oceanic upwelling, but a coastal upwelling, keeping this area very productive in terms of phytoplankton and subsequently zooplankton. And that's the reason why we have sardines here that are very dependent on the plankton, on zooplankton. And later on, I'm going to show you that the enrichment or the productive waters here actually is stretched down to southern Zamboanga Peninsula and even down to the Sulu Archipelago. I'm going to show that in a little while. So we can, by just rough extrapolation, we now know the number of copepods that are found in the stomach of one sardine, and somehow we know the concentration of copepods in the area. We can actually compute the number of sardines individuals that are expected to grow or to be found in that area. No? But these are just simple rough calculation. If you're interested, I'm going to share this to you. Okay, so yeah, those examples actually will give us some appreciation on looking at studying at zooplankton, at the same time their importance to the ecosystem. No? And my main concern is actually the ecosystem service on food or fisheries. No? So I just want to show you that we have these major fishing grounds of sardines. No, we have 6 major fishing grounds of sardines. And that is because these areas have biological enrichment phenomenon, as an example, due to, for example, to coastal upwelling in northern Zamboanga Peninsula. And also, there's another phenomenon in straits. No? Because of the strong currents that enter through straits. So, for example, San Bernardino Strait, Surigao Strait, that would actually also trigger to biological enhancement. So bloom of phytoplankton and then production of zooplankton. Okay, and proof that our zooplankton being linked to sardines are also linked to our tuna. You know, just to show you, this is when I bought the juvenile yellowfin tuna from the Dipolog market. Dipolog, as you know, is in northern Zamboanga Peninsula. I asked the lady to open the stomach, and we saw 4 individuals of Sardinella lemuro in the stomach of our juvenile tuna. We know our different species of tuna actually converge to the Philippine archipelago as juveniles to actually feed, and then they go everywhere towards the eastern part of the Philippines and some on the eastern, but mainly to feed. So basically, if we take care of our zooplankton, we take care of our sardines, and we also are helping our tuna, different species of tuna. Okay, so if some of you might actually read some papers that, okay, yeah, we can use chlorophyll A concentration probably through ocean satellite colors as predictor, but in general, I think that is not, I mean, we seem to getting some issues and problems of just mainly basing on chlorophyll A concentration in relation to fisheries, to fish catch. And of course, other complications, other uncertainties also come in. In fact, we do not have a very good, accurate estimates of catch. I don't know why, and also even fishing effort. And we don't know much about food webs, marine food webs. Dr. Villanoy and I and Dr. Campos submitted a proposal to DOSTP card, and up to now, we have not heard from it. Almost 2 years already. I don't know. Maybe it's not really a priority. Probably, I don't know when we hear from them if our proposal will be funded. But I think that proposal is quite important. So the reason the less the weak correlation between chlorophyll A concentration and fisheries might actually be helped or solved if we focus on zooplankton biomass instead, no? Because I've shown you earlier copepods, no? Zooplankton are directly eaten by sardines, no? So probably if we have a good map of zooplankton in our country, an atlas of where our zooplankton are actually concentrated, probably we can improve further in predicting fisheries biomass. And then if we have a good idea on where and how much of the fisheries biomass would be located in the country, then we are more comfortable with managing them, or thinking of ways to make our fisheries sustainable. So good information on fisheries biomass would actually be needed. No? But how can this be done? Probably one of you there interested want to work on, want to do their PhD on this. No? How can we use zooplankton biomass as a good predictor of fisheries biomass? No? This is already done in North Atlantic. Now, our European friends have they have all the resources and money to fund such study. But a long time ago, they've already started collecting zooplankton data. No, these lines here are actually tracks of continuous plankton recorder. They call it CPR. Uh, I'm going to show you the picture of a continuous plankton recorder. No? So this image here, you can see my cursor. This is it's composed of, it has a metal frame. Inside will have rolled gauze, no, or a bandage ba, no. And as this one is actually towed, no. You see here the continuous plankton recorder is towed by a boat. The scientists call the boat as ships of opportunity. And then as the boat travels transatlantic, the CPR collects zooplankton. And within this, the insides here actually contains formaldehyde. So as the gauze rolls, the zooplankton there, copepods mainly, are actually preserved by formalin. And I think Korea now has developed a similar gadget, no, more improved design. I forgot to show the. But I spoke with a Korean scientist. They said they have a good system. In fact, they have a remotely operated or self-automated plankton collector. No? So this CPR was made in 1931, and it is still being used in Europe and in Australia. In fact, Australia has mapped zooplankton biomass all over the continent with the help of a CPR, continuous plankton recorder. So, okay, so with CPR data and phytoplankton data in the North Atlantic, they were able to develop a model. They call it suitable habitats. No? So by mathematical, you need to have a good appreciation of mathematics also. No? If you want to go into this study, now you link values of phytoplankton and zooplankton biomass, and you come up with a map. Where is the most suitable habitat of zooplankton? And then from there, we can incorporate this information, this database, to predicting fisheries. Now, so the more yellow, so this is not political, huh? Hindi siya dilawan because of dilawan. The more yellow, the higher concentration of zooplankton in the North Atlantic. And they found out that fronts or oceanic polar front tend to be associated with higher concentration of zooplankton, mesozooplankton. So good for them. no, they can further enhance their fisheries management with this information. I hope we can also do that here in the Philippines. And then this is published last year, scientific reports. And one of the interesting statement, generalization that they put in the abstract was that somehow for the zooplankton feeders, so for our fish feeding on zooplankton, for them to be uh, for there is a need for mesozooplankton and subsequently the fish, a specific period for primary production or for chlorophyll maximum production to happen. Otherwise, if of course, it's understandable, no? If the period of primary production is very short, uh, the short period will not match community production of mesozooplankton communities. So there's an ideal period for the phytoplankton to be in the environment so that they can be grazed or they can be fed upon by our zooplankton, and for an ideal community of zooplankton to thrive, and subsequently for an ideal biomass of fisheries that will be supported. Okay, so somehow with that finding, I then thought that maybe it's not available in the NOAA website, you know, the Ocean Color website. No, maybe if we count the duration of maximum chlorophyll A availability, or meaning maximum availability of phytoplankton in the Zamboanga Peninsula, you know, so if we can monitor from January to December and then for several years, the duration of the presence of maximum or even the different levels of chlorophyll in Zamboanga Peninsula, uh, we can somehow this is the information would be very informative to our fisheries, because if as I already as the findings of scientific report study says that there has to be an ideal duration, otherwise our zooplankton community will not thrive. Then if there will be poor zooplankton availability, then our fish will be having problems with their food. So we looked at the ocean color, no, representing chlorophyll production in Zamboanga Peninsula and Sulu Archipelago. So I want you to focus on this region only. As I mentioned earlier, and as discovered by Dr. Villanoy and his team, the northern Zamboanga Peninsula has a seasonal maximum production due to coastal upwelling. So sometime in January, February, we get this maximum chlorophyll production. No, so red is the highest, yellow, then green, and a light blue would be very low levels. No, and by June, this primary production in northern Zamboanga Peninsula already disappeared, or because of the slowing down of the northeast monsoon. No? So the coastal upwelling here is actually driven mainly by uh, coastal upwelling. Okay, so we did that. We counted the for in 1 month we counted the different colors, just frequency, just counting how many in how many within the 2 months. For the whole year we counted how many reds, how many yellow, how many greens, and how many light blues for every month. And the sampling is actually bi-monthly, and these are all sampling from the NOAA pictures or NOAA data. And this is what we found. This is not yet published, no? So somehow there seems to be a variation in durations in the with the presence of the reds, no? So maximum, or the reds usually appear January, the peak January or December, January. But sometimes the presence of the red is very early. October, we can already find the red colors up to March. But sometimes also the red happens middle of December pa, no? Or and then will end in March and then disappears. And we notice that if the duration, the presence of the red colors are longer, it tends to have somehow the biomass of sardines, from we overlaid the sardines landed catch data in our data. If the longer duration happens, then higher biomass of sardines. But again, this needs further analysis. This needs further study. I'm not making any general statements here yet. Because there are other systems in the Zamboanga Peninsula. This is a picture, sea surface temperature profile. You notice here that okay, red is very hot, yellow is cooler. There seems to be in the Sulu archipelago, there seems to be sustained cooler waters, no? And that pattern seems to support continuous high levels of chlorophyll in the Sulu archipelago. Now, you notice that January, February, we have these red, highly productive upwelling events in northern Zamboanga Peninsula. But the rest of the year, nawawala na yung upwelling na phenomenon. But you notice the reds and yellows in the Sulu archipelago are still present. Now, this was actually brought to us by Dr. Villanoy 2 years ago. So somehow, this sustained primary production in Sulu archipelago is also helping our sardines in the Zamboanga Peninsula. And this is part of our proposal submitted to PICARD. We will focus on this region in Sulu archipelago. We want to know what zooplankton and the biomass of zooplankton, phytoplankton, and so on in this area. So this phenomenon here in Sulu archipelago can partly be explained because there are different circulation patterns and movements of hydrodynamic events in Sulu Sea interacting with the Sulu archipelago, keeping maintained primary production in the Sulu archipelago. And by the way, Sulu Sea has sometimes seiche, these are movements, horizontal movements of waves from Palawan to the Sulu archipelago, which can also add to the enhanced primary productivity in Sulu archipelago almost year-round. But not as intense as the coastal upwelling event in northern Zamboanga Peninsula. Okay, so I'm now in my last slide. I think everybody's happy. So please wake up now. So basically my summary is that with zooplankton, with plankton at the base of our ecosystems, our food webs and food chains. So for millions of, maybe hundreds of thousands of years since Philippines became civilized, or when Philippines became Philippines, with people dependent on our oceans and seas, because we know human beings are heterotrophic organisms. We depend on food, otherwise we die if we do not have food. So our country actually will not exist without food, and our country is mainly marine. Always, I want to emphasize that we are actually we define our country now as not just an archipelago with 7,000 islands, but actually our country is a very big body of water with 7,000 islands. Okay, I hope you will be convinced with that definition. We have 230 million hectares of water and only almost 30 million hectares land. So our Philippine territory should be defined as a very big body of water with several islands. Okay. And then, of course, we are at the apex of the Coral Triangle, center of marine biodiversity. Hang on. Yeah, so compared to the water part of our territory, we only have this 7,000 land structures. No, we have two main islands, Luzon and Mindanao, and the other islands here. But if we consider our exclusive economic zone, our country is actually 88.5% marine. So we should even expand our coast guards. We need to recruit more coast guards, and we need to have coast guard vessels. We need to expand our navy to protect our territory. No? So para hindi tayo binubuli palagi. No? Sometimes I feel very angry, but no, I don't want to talk some more. So our maritime ecosystem, our maritime environment actually supports 62 coastal provinces, 61 coastal cities, 832 coastal municipalities. And 60% of our population, no, 55.3, are dependent on coastal areas. So indirectly, they're dependent on zooplankton or on plankton. And in terms of our blue economy, these are the dollar signs of each ecosystems. No, I want you to look at coral reefs. We value them as 915 billion US dollars. No? That's how precious our coral reefs are and other marine ecosystems. And according to Dr. Asansa, academician Asansa, and the other co-authors, our blue economy is actually worth 15 trillion peso. That's of money. Our money is peso. So 15 trillion, no? And our national budget is only 4 trillion every year. So imagine that, no? If we focus our energy and money resources on the blue economy, we can be super rich, no? We can expand, widen our national budget every year. So that's something to think about for our people in the government, people in governance. No? And with that, thank you for listening. So many people to thank, our Japanese colleagues, Dr. Ronald Villanueva, Dr. Campos, Willie Campos, Dr. Cesar Villanoy, soon to be Dr. Christy Acabado, the Philippine Plankton Society, my university, Mindanao State University, Iligan Institute of Technology, the National Research Council of the Philippines, who funded my very first research study in the Philippines on Iligan Bay coffee pods. No? So thank you to NRCP for inspiring my, helping my research career. And that's all. Thank you.
Yay! Thank you so much, Sir Ipe, for that very insightful talk. As usual, we are all entertained with your Tito vibes. I like it. By the way, please send in your questions through this Q&A chat box. And then also we are getting some questions from the FP Live. Thank you so much for joining us. We have questions here right now. Okay, just a moment.
Okay.
Sorry, Pe, are you still there?
Yeah. Okay.
Okay, sir. We have very good questions coming in right now. We have one from Miss Carmina Masikat from Quezon City. She says, thank you. It's a very informative and interesting talk. And her question is, can you elaborate on how the stable isotope feeding analysis experiments were done? Is it possible? Oh, sorry. Yeah. How are the stable isotope experiments are done?
Okay. So stable isotopes are actually present in all living systems. No, so carbon and nitrogen stable isotopes are present. These are heavier isotopes. So all organic matter in this planet will have stable isotopes. So somehow, uh, for example, we eat rice, no, every day, and rice itself has stable isotopes, no. And once you ingest the rice, they will be assimilated, incorporated in our muscles, everywhere. No, the carbon and nitrogen component of the rice will be incorporated, and no, actually carbon, basta carbohydrate, carbon ang marami dyan. So more carbon stable isotopes. And then if you eat fish, if you eat meat, nandun naman yung source ng stable isotope ng nitrogen. So once they're incorporated in animal's body, they become part of the body. And then we measure, we collect tissue samples, no, muscle samples, and then measure the amount of carbon and nitrogen stable isotopes. And we also measure the nitrogen and carbon isotope from the potential food or potential prey of that particular species. And then by specific equations, calculations, we can then establish the link between the feeder and the food in terms of carbon and nitrogen stable isotope. And you need a specialized, very expensive machine to analyze carbon and nitrogen isotopes. You use a mass spectrometer and an elemental analyzer together to actually measure the amount of stable isotopes. And for us, it is more accurate in understanding the food chain or the link between different species in terms of feeding. No? And yeah, so basically that's it. I hope I have explained it properly.
Thank you, Sir Ipe. We have a very interesting question here. How different will the plankton profile be on the eastern parts of the Philippines? Most studies presented today are mostly on the western part. How about the parts facing the Pacific?
Oh, okay. So very interesting. No, in terms of okay, I'll show you again a picture. No, I've shown earlier. Okay, where is my? Okay, so yeah, just to show you, where's my? Hindi ko na makita yung aking.
Share screen button.
Ah,
Andiyan na po, sir. Nag-stop na ng sharing dito.
hindi ko na makita yung aking PowerPoint. But perhaps some of you have caught a slide there showing the Philippine map with different colors. No, you notice that on the eastern side of the continent, medyo ano siya, no, blue siya. But on so that means halos buong taon, no, mababa yung dito sa Pacific Ocean na side, except sa ano, except yung malapit sa islands, no, for shallower waters, mga embayments close to the islands, doon ang mataas ang plankton production. But as you go deeper sa Pacific Ocean, mababa talaga yung plankton production. Um, and also except sa kasi dito sa Pacific Ocean meron tayong ano, 'di ba? Equatorial currents. We have the North Pacific Equatorial Current and the South Pacific Equatorial Current. And this is moving towards the Philippines from the east to the west. So somehow dito sa area na may gumagalaw na current, yung equatorial currents, nagbo-bloom dyan yung mga phytoplankton din, no? So they can be carried towards the Philippines. And the mechanism is also upwelling. And but generally, mababa yung plankton production sa eastern portion, no, of our territory compared to the west. Yun lang po.
Thank you, sir. We have.
Hang on, Christy, kasi andito na siya eh. Can you
Oh,
see
yeah.
that?
Yes.
Okay. So you notice na yung blue, the eastern portion of the country, blue siya, no? So mababa yung plankton production compared to our central Philippines, Visayan Sea, Bohol Sea, medyo green siya. And also the Sulu and West Philippine Sea. I don't want to call it the other way. Alright? So West Philippine Sea and Sulu Sea. So meron talagang difference sa Eastern Philippines at saka dito sa western part of the country. Yan lang.
Oh, okay po, sir. We have a question here from, sorry, I cannot find the name anymore, but the question is, can you identify potential zooplankton hotspots in the Philippines? We have our favorite place.
Yeah, partly we already have some idea. Pero as I said earlier, we need more zooplankton studies. No? Okay, so ano ba yung hotspot? I will show you again. Saan ba yung screen share? Okay, so mga hotspots depend. It also depends on time. It depends on what period of the year. Yung inaral namin ni Christine na Dr. Campos, ni Sir Mac Malugaw, yun nga, Northern Zamboanga Peninsula, marami ditong zooplankton, phytoplankton pag peak season during upwelling season. So probably December, January, February, marami dito. And as I've said also, dito sa Sulu Archipelago, marami din dito sa mga strait area, yung mga bukana between dito sa San Bernardino, Surigao Strait. Dito sa Coron, malapit din sa Mindoro, depends on the year also. Marami ditong zooplankton din, Northern Palawan. In fact, this area here is a spawning ground ng ating mga galunggong. And galunggong also are zooplanktivores, no? Hindi lang sardines, kumakain ng zooplankton. Saan pa ba? You notice also dito sa Butuan Bay, pula siya, di ba? That means marami ditong plankton din, zooplankton, but this area is different. Usually itong mga may malalaking ilog like Agusan River, they bring nutrients to the bay, and the nutrients here will trigger the bloom of phytoplankton. So hindi siya upwelling, but actually estuarine, no? Estuarine plume siya. So in areas na may malalaking ilog, malinis yung tubig, may plankton dito. You notice also Manila Bay, pumupula siya, no? Baka red tide ito. No, actually, Manila Bay, marami pa rin siyang zooplankton, pero sa upper layers ng water, no? Kasi the bottom waters of Manila Bay is already almost hypox siya, no? O wala ng oxygen. But probably in the upper 50 meters, 60 meters ng Manila Bay, marami pa rin zooplankton, kaya marami pang, ano ba sa Tagalog yung tamban? Ha?
Same lang po,
Yung,
sir. Tamban
ha?
din.
Tamban
Tamban
din
din
ano?
po. Mm.
May ano pa rin dito eh, Sardinella gibosa at saka Fimbriata. So, may mga zooplankton pa rin dyan. Dito rin sa Catanduanes area, sa Bicol, Northern Luzon, sa Lingayen. So actually maraming areas pero konti lang yung nag-aaral dito, no? And we hope that more Filipinos will be encouraged to study on zooplankton.
Okay, salamat, sir. We have a question here from Vaileni Encarnacion. Do you also identify invasive species of zooplankton?
What is that again, Christy?
Encarnacion, I'm not sure if I read the name right. Vaileni?
Ano yung question?
Do you also identify invasive species of zooplankton?
Oh, invasive species. Yeah, we studied one in Lake Lanao. So, but Dr. Ray Papa, he will be the second speaker. So, he's familiar with invasive species na zooplankton. Most of our zooplankton species are cosmopolitan, no? So they can be found in other regions in Indian Ocean, in the tropical belt of the planet. Halos pare-pareho yung mga species, may commonality. But there are also unique species in the Philippines. We need to understand all the species of the country before we can say na may invasive, no? So if we know something na hindi typical sa Philippines, then that can be invasive. Pero invasive siya, kainin ka agad siya ng mga isda natin, no? We have so many fish na pwedeng kumain sa kanya.
We have a lot of questions coming in, but we are also running out of time. So I'm sorry if hindi po natin ma-ask lahat. But I will have one last question from the audience. Okay, maybe this is also similar to my question because we want to promote the study of planktology to younger students. So the question is from an anonymous attendee. Is it possible for our scientists to study the zooplankton profile in the WPS? Do we have chances to get funding to do researches in the WPS?
Oh yes, no, very big chance. No, I'm very hopeful, very positive, because right now UPMSI was able to get funding to buy 5 boats, 5 research vessels, including equipment. No, in fact, on Friday, we will have a meeting with UPMSI, with Dr. Deo, Dr. Laura David, and we're going to discuss on we have the money to buy the 5 boats, and I think 2 boats will remain in Luzon with UPMSI. 1 boat will go to UP Niagao, UP Visayas, and 1 boat will go to UP Tacloban, and 1 boat will go to MSU IAT. So yun yung 5 barko. And once the boat will be available, we also requested the government to equip the boat. So it is a research vessel. So we call it the NAR fleet, the National Academic Research Fleet. And once we have all the 5 boats, we can, of course, study everywhere in the country, and most especially to our West Philippine Sea. Now, in fact, our president has encouraged more presence and activities in West Philippine Sea para hindi naman siya naaagaw ng ibang mga bansa dyan. So, yes, we can study. In fact, later on, we will be requesting volunteers or people who would like to join in the research team. No, so you can join in as a research team and work on zooplankton of West Philippine Sea. No, so marami pang dapat aralin natin how productive our waters are, specifically our territory at West Philippine Sea. Okay.
Okay, thank you so much. Again, I would like to apologize for the other questions that we cannot read today. So I think we can move forward to awarding the certificate for our lecturer today. First of all, but before that, I'd like to thank our partners again, the Institute of Marine Fisheries and Oceanology, the director, Sheila Santander de Leon, Jonalyn Mateo, our admin officer, Helen Monteclaro, DISP, UPV, Sir Paeng, and the Plankton Society of the Philippines. Yep. Our certificate for Dr. Ephrime Metillo is awarded to him in recognition of his outstanding contribution as keynote speaker of the presentation entitled Philippine Zooplankton from Wanderers to Ecosystem Drivers at the first lecture series on Philippine Planktology on April 21st, 2021. Signed by the director of IMFO-CFOS UP Visayas, Plankton Society of the Philippines Vice President, and the administrator of the Philippine Planktology FB page. Thank you so much, sir, for this pro bono lecture. I know that you are doing this also with the same cause as our group. Thank you, sir.
Thank you very much also for the invitation. Thank you.
Yeah, thank you. I would like to say that this is a very successful first lecture. We were able to have over 300 Zoom participants and then 64 on FB Live viewers. I would like to thank everyone for coming and joining us in our lecture. And I would like to acknowledge the youngest participant in the audience, my friend Izor. And then, yeah, okay, so thank you so much for attending. Please answer the evaluation form that will be emailed to you. And I would like to see you again next lecture on May 10. Again, at 2 p.m., please visit our Facebook page, the Philippine Planktology, for more updates. Thank you so much and see you again next time.
Chrissy, I have to go. Thank you. Maraming salamat.
Thank you, Sir Ipe.
Okay, bye-bye.